In our first video for D1.2, we looked at the process of transcription, the first step of making proteins in this process of protein synthesis. And in the second video, we'll look at primarily the process of translation, using that product of transcription to actually produce a polypeptide that will become a protein. Translation is the second step of protein synthesis in which the amino acids are linked in the correct sequence in order to form a polypeptide. And that chain of amino acids is made from some combination of 20 different amino acids in various sequence and amounts or lengths. And the code or the instructions for their putting together, for their linking, is provided for or provided from the messenger RNA that's made during transcription. And if you remember, ultimately that messenger RNA is coming from DNA, so DNA is providing the instructions to link those amino acids in the correct sequence to make the polypeptide. Translation as a process takes place in the cytoplasm, and within eukaryotes, this occurs after the transcriptional modification that takes place within the nucleus, so the removal of the introns, for example. In prokaryotes, because prokaryotes don't have a nucleus, this occurs directly after transcription, and there's also no modification of the messenger RNA within prokaryotes. There are three main molecules that are needed to synthesize the polypeptides to make that protein. The first is messenger RNA, and this provides a binding site for the ribosome that can be read one codon at a time, so the ribosome actually binds to that messenger RNA, and so it reads it or looks at the messenger RNA 3 nucleotides at a time. That's referred to as a codon, and that codon indicates a specific amino acid that should come next in the sequence, as well as where to start the process, we call this a start codon, and then where to end the overall process, called a stop codon. That messenger RNA can be used multiple times. It's not a single use, it can be used multiple times. The second molecule is tRNA, and this stands for transfer RNA, and it is used to transfer amino acids to the ribosome, and it does so by matching the anticodon, so it's another 3 RNA nucleotides that are found on this tRNA molecule, and they're complementary to the codon that's found on the messenger RNA. And they must be matching, or they must be complementary in order for that amino acid to be delivered. The tRNA molecule has a distinctive shape that's recognized by an activating enzyme, so that only one particular amino acid can be put on to the correct transfer RNA molecule. The third molecule that's essential for this process is the ribosome, and this is the structure that actually does the synthesizing of the polypeptide. It's made of 2 different parts. One is the large subunit and one is the small subunit. It kind of looks like a hamburger bun if you put the 2 pieces together, with the messenger RNA kind of being whatever is inside of the bun is sandwiched in between them. The ribosome has a binding site for messenger RNA on the small subunit, so that's where it binds. The large subunit has 3 binding sites, E, P, and A, and those are for the tRNA to be able to connect or bind to. And that's where those peptide bonds between the amino acids will be formed at the large subunits as that transfer RNA delivers the amino acids, as we'll see here shortly. So again, the codon are 3 nucleotides on the messenger RNA that the ribosome reads one codon at a time, and they match with or are complementary to the anticodon that's found on the transfer RNA. And the complementary matching of the messenger RNA and the tRNA, the codon, the anticodon, that's essential to make sure that the correct sequence of amino acids are bonded together. And without this, then the sequence would not be in the correct formats to produce the correct polypeptide and protein. The genetic code within DNA is really providing the instructions for the linking of those amino acids together to form a polypeptide. As we know, there are 4 different types of bases within DNA nucleotides: adenine, thymine, guanine, and cytosine, and there's a total of 20 different amino acids that organisms are using to be able to produce these polypeptides. And so the code, using only 2 DNA bases, if the code or the instructions that's codon on the messenger RNA, if it was only 2 bases, that only gives us 16 combinations. That's not enough to match all 20. And so we see a triplet code, this codon, of 3 nucleotides on the messenger RNA, ultimately coming from 3 DNA nucleotides that are used to be able to provide the instructions for linking of those amino acids. And so then this yields in total 64 different combinations of these 3 of the 3 bases of adenine, thymine, guanine, and cytosine. There's 64 different combinations of how those can be put together. And of all of those, all but 3 of them code for a specific amino acid. those three coding for a stop codon, so that's like the off switch to turn off this translation process. Some codons code for the same amino acid, so there's some duplicates, some overlaps. Some codons code for the same amino acid, and this is referred to as degenerate, in which multiple of the same sequence or different sequences of codons can call for the same amino acid. A specific codon can be identified from a messenger RNA sequence by using a messenger RNA amino acid chart like we see here. And so essentially how you use this is you start with the first nitrogen base that's in this codon, A, G, U, because it's RNA, or C, and then there's multiple different types, but in this circle-orientated one, you move to the next circle, identifying the second nitrogen base and then the third nitrogen base. And this allows you to pretty quickly and easily identify the specific amino acid that a different codon calls for. And you should be comfortable using this chart, so maybe a little bit of practice would be beneficial so that you can identify amino acids based off of the mRNA codons. This code is universal, and it's used by all living organisms as well as viruses because these living things have DNA, and the process of transcription is similar in all organisms. And so we see these amino acids and this process be used by all different types of organisms. The initiation of translation is really begin even before the linking of amino acids takes place. And it starts with making sure that the correct amino acid is on the correct transfer RNA, as previously mentioned, because if the incorrect amino acid is on a transfer RNA molecule that has not the correct anticodon, then because the messenger RNA is calling for a specific transfer RNA, if that correct amino acid isn't attached to that transfer RNA, then it would mess up the entire sequence and thus the polypeptide and the protein. So in order to ensure that the correct amino acid is on the correct transfer RNA molecule, an activating enzyme that has an active site that fits the transfer RNA molecule, the tRNA actually binds to it in a specific amino acid then is attached. That amino acid and the transfer RNA is determined by the shape and the polarity, so that the anticodon that matches the messenger RNA codon will receive the correct amino acid. And this ensures that that process is correct. Some energy is required in order to make this process take place, and that comes from ATP. Once the amino acid is on its specific transfer RNA, then it can bind to the P site of the ribosome, and this is on the large subunits. That first amino acid is always coded for by the start codon AUG. That's the start codon for all different types of polypeptides, which is the amino acid methionine. Secondly, in this process, then another transfer RNA with potentially a different amino acid, most likely a different amino acid, then binds to the A site of the large subunits, and a polypeptide bond is formed between the two amino acids, the amino acid that's at the P site and the A site. A bond is formed between them, polypeptide bond. The tRNA that's at the A site then releases its, at the P site releases its amino acid, and then the two amino acids are linked by a peptide bond that is attached at the A site. The tRNA then that was at the P site moves to the E site, and essentially everything shifts down. The ribosome moves in one codon, and that causes the tRNAs to shift down as well. The transfer RNA of the first amino acid, the methionine, is at the E site and is ejected. It's released from the ribosome, thereby shifting the tRNA that was at the A site to the P site and allowing that process to be repeated. And so it's just a continual cycle until eventually the messenger RNA codon that is a stop codon is reached, which thereby then ends this translation process. Because both DNA and RNA have a specific direction, because both ends of the molecules are different, the phosphate that's attached to the carbon atom, the carbon atom number 5 of the sugar, is the 5' end. That signifies the 5' end. And then the open carbon that doesn't have something bonded to it of the sugar, the open 3 carbon, signifies the 3 carbon end. And so transcription, very similar to DNA replication, The RNA nucleotide is added at the growing end of the strand, which is the 3' end, and again, that's similar to DNA. And in order to form this messenger RNA, covalent bonds need to be produced or created between the sugar and the phosphates, and that occurs between the 5' and the 3' end, and that also uses the energy source of a nucleoside triphosphate. Translation, then, very similar, also moves along the messenger RNA towards the 3' end of that messenger RNA. And so both transcription, translation, and even DNA replication are all moving in the 5' to 3' direction. tRNA as a molecule has, or sometimes referred to as a clover leaf shape, in which the amino acid is located on the opposite end of the anticodon, if you're looking at the structure of it. And so in the process of translation, the tRNA with the amino acid is going to bind to the messenger RNA that's on the small subunit of the ribosome. This complex binds to the 5' end of the messenger RNA and then slides along until it finds the start codon AUG, which would be complementary or have an anticodon of UAC. The linking of messenger RNA and tRNA is carried out by hydrogen bonds, just as we see in DNA molecule. And once that start codon is identified, the large subunit then binds to the small subunit, and that tRNA molecule, that start codon tRNA molecule, is at the P site. It's located at the P site. The next site on the ribosome, the A site, then allows for a second tRNA molecule to bind. That's complementary to the next codon on the messenger RNA, thereby allowing a peptide bond to form between the two amino acids, and the amino acids held together by the peptide bond are attached to the tRNA at the A site. The binding of the amino acid to the tRNA at the P site is released, so it's only attached to the A site. The ribosome then moves from a 5' to 3', one codon at a time, where the P site tRNA is moved to the E site and ejected, and the tRNA that was at the A site gets shifted to the P site, and thereby allowing a new transfer RNA molecule to bind and continue the process until a stop codon is reached. The polypeptides that are produced during the translation process must be modified to become a functional protein, and there's a couple of main types of changes. First is the removal of the methionine from the 5' end of the polypeptide. That was our start codon. There can also be alterations to the side chains of amino acids. For example, the phosphorylation or addition of carbohydrate molecules to those side chains. Oftentimes also the polypeptide is folded and makes intermolecular interactions for stability. This produces tertiary structure, where we see interactions between the different R groups, for example, disulfide bonds between cysteines. There can also be the removal of part of the polypeptide chain, or the combining of two or more polypeptides or non-polypeptide components, and this would then produce quaternary structure, and is an example that we discussed earlier in the unit of the course for hemoglobin. Insulin provides a good example of these different modifications because the insulin gene, INS is the abbreviation, is found on chromosome number 11, and it's transcribed in the pancreatic beta cells to produce messenger RNA, which then produces proinsulin with about 110 amino acids. This then passes into the lumen of the rough endoplasmic reticulum, and at that point then there's a removal of about 24 amino acids by protease enzymes that recognize 3 adjacent alanine amino acids in that overall sequence, and it leaves the molecule proinsulin, which is a chain of 86 amino acids. So in this first step, we're removing some of those amino acids, making it a smaller chain. That proinsulin then becomes folded with 3 disulfide bonds, and then there are some peptide bonds that are broken by proteases through the recognition of the positively charged amino acids, lysine and arginine. And then this removes 33 amino acids in total. And so this produces 2 separate chains, but they're linked by those disulfide bonds, where the first chain, A chain, has 21 amino acids, and the B chain has 32 amino acids. Lastly, there's a pair of amino acids removed from the B chain to produce the final mature insulin that has 51 total amino acids. So we started with 110 amino acids and end with 51. So half of it's removed, and the shape is completely changed into having these two chains together. And so this is a good example of how much that polypeptide sequence can be modified to produce the final mature protein. Proteins generally have a pretty short lifespan, and that's due to the different activities changing that make maybe the protein unnecessary. The protein structure itself can be easily changed by free radicals or other chemicals. And the protein can become misfolded or denatured, as we've seen in other units in the course. Non-functioning proteins, so after they've become unused or they're not functioning anymore, can be broken down and recycled by proteasomes. Identified proteins are tagged with a chain of small proteins called ubiquitin. Tagged proteins are then unfolded and moved into the central chamber of this proteasome by a subunit that's oftentimes powered by ATP. Active sites of multiple proteases face the central inner chamber or channel, and then they break the proteins into short amino acid chains. And then sometimes additional digestion takes place in the cytoplasm, thereby allowing those amino acids to be freed up and used in the construction of new proteins. So it helps to recycle the reuse of those amino acids. As we've seen, mutations can have big impacts on proteins, and a mutation, again, is a change in the sequence of a gene. Base substitutions are the most common and usually result in one amino acid being changed within the polypeptide product that can have sometimes massive effects and have big impacts. And sickle cell disease is a common example of this. It's the most commonly inherited condition in the world. Unknown exactly how often that mutation occurs. It's very common or more common in parts of East Africa, where about 5% of the babies have 2 copies of this mutated allele or form of the gene, and they are very anemic. As we'll discuss here, about 35% have 1 copy of this mutated allele. And we typically see sickle cell anemia more common in places that have high rates of malaria, because people who carry the sickle cell allele, this mutated form of the gene, generally have less forms, severe forms of malaria. And so the mutation that causes this disease is thought to maybe have originated in areas where malaria is common, because it actually provides some protection from the disease of malaria. And so this sickle cell disease is an example of a single base substitution disease. HB is the symbol in which the allele HBA is the most common and the non-mutated form of the gene. The mutation of a GAG to a GTG in the DNA produces the mutation, and that's signified by the HBS allele. The mutation can be inherited by offspring if it occurs in sperm or egg cells. And so the mutated form of the allele, the HBS, that mutation occurs in the 6th codon and has GUG instead of GAG. And so what that means is it's a valine amino acid instead of a glutamic acid. And that change affects the hemoglobin molecules when tissues are have low amounts of oxygen, when there's low concentration of oxygen, and it causes the molecules to stick together, and it forms chains that have this rigid bundle structure that kind of looks like a sickle. And so that's thus the name, a sickle cell shaped, a sickle being like a tool that farmers would maybe use to chop down hay or grasses or something like that, kind of a half moon shape. And that sickle cell, those cells, then can become trapped in blood capillaries. It blocks and reduces the blood flow, and it causes damage to the tissue, and it's also painful. When the oxygen levels return to normal, the cells break up and return to their normal shape. But this process is repeated, and it produces damage within the hemoglobin and the plasma of the blood, and can also cut down the life cycle, how long those red blood cells persist, to only about 4 days. And so that can lead to anemic conditions where the body can't produce red blood cells fast enough to be able to keep up. And that's all caused by this single base substitution. That's what's causing this disease to persist. And so it's an interesting case study of one of the many different types of genetic mutations that can cause genetic disorders and diseases.